these are notes on genetic transformation. So it's going to kind of walk you through the main parts of the procedure, and I'm going to show you kind of what you do, so really looking at what it is you're going to see, and then what the bacteria does, so that you can see like what's going on at the level that you can't see. All right, so first, just a little overview of what's going on with bacterial DNA and plasmids. So here we have E. coli. E. coli is a bacteria, so it's a single-celled organism, and it has a circular ring of DNA. So unlike eukaryotes, which have DNA inside their nucleus, prokaryotes, which don't have a nucleus, have a circular ring of DNA. So this thing here is the E. coli DNA. A plasmid is a circular ring of DNA where you've put certain genes on it that you want, okay? So we have a plasmid that glows red or possibly orange, I can't remember which color I ordered, and is resistant to an antibiotic called ampicillin. So ampicillin normally kills E. coli. E. coli is not resistant to ampicillin. But what we're going to do is we're going to insert this plasmid into the E. coli. So this, by the way, is the entire E. coli cell membrane. We're going to insert the plasmid in there, and then the E. coli will also start to make the proteins of the plasmid. So go back to your knowledge of how traits are expressed, right? The cell reads the DNA and it translates it, or transcribes it and translates it, and then you get these proteins. So the E. coli will start to produce the proteins of the plasmid. So it too will be ampicillin resistant and glow red. So that's the overall idea of what we're doing. So here's what we're doing, and again, the written directions give you far more detail, so I would almost have those out so you can kind of follow along as you're going through kind of what the pictures look like and what this feels like to the E. coli. And many thanks to our UW observers, Mr. Putman and Mr. Taylor, for helping out with the pictures. So you're going to first scrape an E. coli colony. So you can't see individual cells, they're far too small, but I'm going to grow a bunch of colonies, and this is my pretend agar dish with, you can see little dots on it. We're going to pretend those are E. coli colonies. So you're going to take a toothpick to scrape up one E. coli colony, and they're going to look just about that big. They're tiny. But in that, remember, we're talking, I don't even know, bajillions of cells. And then you're going to put it in your microtube where you've already placed the calcium chloride. And whether you end up placing the calcium chloride in the microtube or I do that for you is still a little bit to be determined. But you know how to use micropipettes to pipette in, I think it's like 250 microliters of calcium chloride. It says it in the directions. And so then you're going to keep your little microtube on ice, and you actually, if you're looking at the directions, you have two microtubes because we're going to do a control tube and a plasmid tube. And what the calcium chloride does is it creates some charges, the positive charges on the membrane of the E. coli so that the membrane doesn't repel the DNA and or the plasmid that we add, which is DNA. But remember, DNA is negatively charged, so if we have some positive charges on the membrane, then the positives and the negatives will attract each other. So then you're going to, of your two tubes, add the plasmid to the pFLO tube only. And again, if you're not reading the directions, you're like, what's a pFLO tube and what does that even mean? But read the directions. So you're adding 10 microliters of the plasmid, and so this is a little reminder on your micropipette tube here that this is a P20 tube, and you can see that would be 10 microliters because this is the tens place, the ones place, and then the tenths place. So you may need a little refresher, and you're going to take 10 microliters of the plasmid and add it to your pFLO tube. So that's what's going on here. And we keep the whole thing on ice. Oh, I thought I had words there. Apparently I have no words. The E. coli is not saying anything because really all it feels like is, hey, here's the plasmid, here's the E. coli, goody. Then we're going to give it a heat shock. So you're going to put it into the hot water bath, and you'll have these little floaty test tube racks, and you put your little microtubes, both of them. We're doing the exact same thing to each one, except that at this step here, we're adding plasmid to pFLO. We're going to add distilled water to the control tube, just so we're adding the same amount of stuff, but the distilled water is, of course, unreactive. So then we give them both a heat shock, and as it says in your directions, the timing of the heat shock is incredibly important, so make sure you have your timer ready to go before you put them in so that you can press start immediately, and it's 90 seconds. Okay, so however you choose to time that, you can use the second hand on the clock in the room, you can use a timer on a phone, you can use a regular timer. It's up to you, but you need exactly 90 seconds. The timing is critical. So you put in, you shock it for 90 seconds, and in the heat, what the E. coli is feeling is kind of like, yikes, what's going on? Why? I was nice and cool and happy. Now I'm really hot, and this is not cool, people. But that's important. We want the E. coli to be a little disturbed at this point, so this is good. So then we put it back on ice. So you'll have a little ice bucket that you've been using the whole time. If I can get crushed ice, I will. Not crazy about big cubes of ice for little tubes, but whatever. They go back into the ice. And now the E. coli is like, no, wait, hang on. Now we're back in the cold. What is going on? So not cool. And the E. coli starts to essentially cast around for information. So I want you, the analogy is, if you are in an environment that is very disorienting to you, maybe you've landed in another country where not only do you not speak the language, but maybe even the alphabet is different, so like you don't recognize the letters, right? And you're going to cast around for any information you can find, probably in the form of a map or a translation book or something like that, right? The E. coli too is going to cast around for any information it can find, and oh look, it finds some information in the form of DNA. And so some of that plasmid then can enter the E. coli cell. Not all of it does. You'll notice not every E. coli cell ends up transformed. So when you say transformed, you mean the plasmid has gone in. Some of it does, some of it doesn't. And so that's a really important thing when you're making your predictions to keep in mind. So then we want to treat our E. coli well. So we're going to give it some food. So we're going to give it 250 microliters. So this is a P1000. So remember, this would be the thousands place. So this is the hundreds place, and this is the tens place. Okay? So this is 250 microliters of luria broth. And luria broth is very nutrient rich if you are E. coli. Okay? And so you add that, again, to both tubes, because we want to do the same thing to both tubes. We're going to give our E. coli some food. And the E. coli is thinking, ooh, I love luria broth. This is my favorite. Now things are starting to chill out. I've got some information. These nice people have given me some food. And at this point, you're actually going to stop for the day. So you have to get this far in a day before you can stop. And then we're going to put our E. coli into the refrigerator overnight. When you then get it, the next, and that again is both tubes, so the E. coli with the plasmid and the E. coli without the plasmid. And the directions are really clear as to when to change tips. Like, so you don't always want to use the same tip because you don't want to contaminate your tubes. I'm talking about on the micropipettes. So then the next day you get your tubes back and you're going to put it back into the hot water bath, but this is a much lower temperature. So this is like nice and cozy. Think of it as a gentle hot tub. So now what you've done is you've shocked your E. coli and freaked it out. You've made it collect information, but then you've given it some food. You're now putting it in a hot tub. it's happy. And so what do happy things do? The goal of every organism is to reproduce. So the E. coli is like, oh, this is nice. I had a nice meal. I'm in a relaxing tub. Clearly it's time to reproduce. And we want the E. coli to reproduce because the E. coli that has been transformed, so like this one here that has the plasmid in it, when it reproduces, it will reproduce with the plasmid. So we'll get lots and lots of that. So then you're going to plate your cells onto your Petri dish, okay? And you're actually going to have 4 Petri dishes. And so on the next slide, I'll show you what you have. But you're going to take 100 microliters and plate that directly into the center of your agar dish. And then you have a choice as to whether you use a spreader. So this is a spreader, and you kind of take the spreader and you would orient, so this is the bottom part of the spreader. You'd orient it like this, and then just spread around once in a clockwise direction, like so. You could do once in a counterclockwise direction, it really doesn't matter. Or alternatively, you could, I've had a friend, the first time I did this didn't spread very well in that I pushed all my E. coli out to the side and it just grew around the edges rather than right in the middle. So I have a colleague who just micropipettes the 100 microliters on and then shakes the Petri dish a little just in her hand, just to kind of spread that around, and that's all she does. And it is your decision which method you want to use. So you have this on 4 plates, and then your E. coli says, and actually your non-transformed E. coli would say this too, but they say, okay, time for asexual reproduction. So the idea here that this is asexual reproduction means there's no meiosis, there's no messing around with, like, you're not increasing genetic diversity any. You're essentially getting clones. You're replicating the DNA and splitting the cell, and replicating the DNA and splitting the cell. So this is asexual reproduction, where you don't have some chromosomes, well, or we'll say DNA here because it's not in chromosome form, some DNA from one parent and some from another, and you get a completely genetically unique offspring. This is a genetically identical offspring. And so once you've plated, you're going to put them into an incubator for overnight, and we'll check in on them the next day, and you'll get to see what grew. And what grows depends somewhat on what you have on your plates. It also has to do with how well you followed the protocol, but some of it has to do with what you have on your plates. And some plates have ampicillin and some do not. So let's look at your 4 plates. So you've got your 4 plates. You're going to have 2 plates that just have LB agar. So the LB is like the Luria broth. So this is a great nutrient for E. coli. And so you're going to put 100 microliters from your control tube on one plate and 100 microliters from your pFLO tube on your other plate. Then you're going to have 2 plates that have the LB, the Luria broth, plus ampicillin, okay? And same thing, you're going to put 100 microliters of the control tube E. coli on your LB and ampicillin agar, and then 100 microliters of the pFLO tube on your LB and ampicillin agar plate, so the 4th plate there. And so what I want you to predict is what's going to grow, if anything. There may be a plate that nothing grows on each plate. And remember that we transformed with E. coli that is going to glow, like I said, I think red. It might be orange. It doesn't matter. E. coli that is not transformed will grow white. So you'll see white specks, just like the E. coli you saw when you scraped off a colony. So when you're thinking of predictions, you're thinking of what color E. coli, if any, am I going to see growing on these plates. And it may be one color, it may be more than one color, you may have a plate where nothing grows. So you have to kind of figure out, based on the genes of this E. coli and the substances on the plate, what will I see grow? And so go ahead and make those predictions. You can make them now, you can make them after you've started the you can make them between day 1 and day 2, you can make them after you plate, before day 3, but you definitely want to think this through to make sure you've got the right ideas. All right, so please on Friday ask a lot of questions so that you're really prepared to hit the ground running on Tuesday when we do this.